So now we need to look at the process of excitation-contraction coupling. And the process of excitation-contraction coupling is the process by which the neuron communicates with the muscle fiber to get the muscle fiber to contract. So there's 4 stages in excitation-contraction coupling. 2 of them don't make it into the name. The first one is stimulation. So we need the stimulation from the neuron, from the motor neuron. Then there's excitation, which is when the sarcolemma gets excited. Then there's contraction, when the sarcomeres actually shorten. And then there's relaxation, which is when there's a process of cleaning up of the ions, pumping them back into the places where they're supposed to be so that the next contraction can occur. So we're going to have 4 stages: stimulation, excitation, contraction, and relaxation. And those 4 are going to break down into multiple steps. So I'm going to color code them. So we'll start with stimulation, and I'll put the steps of stimulation in red. So for stimulation, it starts with an action potential travels along the axon of a motor neuron. So this thing right here is the axon terminal from a motor neuron. And here we're representing the action potential. That squiggle is supposed to represent electricity. So it's electricity traveling along the motor neuron, and it's going to reach this endpoint, which is the axon terminal. So that's number 1. An action potential travels along the axon of a motor neuron. When the action potential reaches the axon terminals, which is this bulb right here, so this bulb is the axon terminal. When it reaches there, acetylcholine is released into the synaptic cleft. So what does that look like? There are little vesicles in this axon terminal, and the vesicles contain acetylcholine. I'm going to put little green dots in here. Those little green dots are the acetylcholine, which is a neurotransmitter. It's a neurotransmitter that makes muscles contract. And so what needs to happen is these vesicles are then going to fuse with the plasma membrane of the axon terminal. And when they fuse through the process of exocytosis, which we learned is a bulk way of dumping some material out of a cell, through the process of exocytosis, that acetylcholine gets dumped into this space here, and the space is the synaptic cleft, right? So this space here is the synaptic cleft. So now the acetylcholine is in the synaptic cleft. So now the acetylcholine has to diffuse across the synaptic cleft, okay? So it's going from high to low. So we know it's higher where it got released, and so it's going from high to low. And when it diffuses across, it's going to bind to receptors that are on the motor end plate. So we're going to put in some little receptors here. Okay, so these are little receptors on the motor end plate of the skeletal muscle fiber. And so the acetylcholine is going to go and it's going to bind. So I'm going to draw like a bigger one just to emphasize how it's binding to receptors. So that's step 3, where the acetylcholine binds to receptors on the motor end plate of the sarcolemma. So acetylcholine binds to, that's what we see there. Acetylcholine binds to receptors on the motor end plate. So all of that, 1, 2, and 3, that represents stimulation. So stimulation includes the action potential traveling along the axon of the motor neuron. When the action potential reaches the axon terminals, which is this foot-like thing, this bulb at the end, acetylcholine is released into the synaptic cleft. The acetylcholine diffuses across the synaptic cleft, and then it binds to receptors on the motor end plate, which is number 3. Okay, so that all of that is stimulation. So we can call that whole section stimulation. We are going to do the next section, which is excitation. So excitation, I'll do orange. All right, so for excitation, when that acetylcholine binds to receptors on the motor end plate, what happens is you end up opening up little sodium channels. Okay, so you end up opening little sodium channels that are in the motor end plate. So I'm going to open, in the motor end plate, I'm going to open up little sodium channels here. And when we open up the little sodium channels, we know that everything wants to go from high to low. So sodium is going to flow in, going from high to low. So there was sodium out here, and it's going to flow in the channel, going from high to low. So there was sodium outside the cell, it's going to flow in, going from high to low. I'll skip that one because we kind of ran out of space, but sodium flows in high to low, okay? So that is step 4. The binding of acetylcholine causes the opening of sodium channels in the motor end plate. And when those sodium channels open and the sodium ions enter, that creates an action potential that spreads across the sarcolemma. So when that sodium enters, you get an action potential that spreads across the sarcolemma of the muscle fiber. Okay, so that's number 5. Action potential spreads across the sarcolemma, and it's going to go down into the T tubules. Okay, so from that sarcolemma, those T tubules, the little holes in the membrane, they bring that action potential down deep into this muscle fiber. So that's step 6. The T tubules bring the action potential deep into the muscle fiber to reach the sarcoplasmic reticulum. Okay, and this thing here that looks almost like a bone with like holes in it, that is your sarcoplasmic reticulum. So at this point, when that action potential reaches that sarcoplasmic reticulum, it's going to cause the sarcoplasmic reticulum here to dump calcium ions into the cell. So again, on this side, we're going to dump calcium ions into the muscle fiber. Okay. Calcium ions get dumped into the sarcoplasm of the muscle fiber from the SR. Oh, and that's step 7, where the calcium gets dumped. That's step 7. So the calcium is getting dumped from the sarcoplasmic reticulum into the sarcoplasm. So 4, 5, 6, and 7, all of that is considered excitation. So that's excitation of the muscle fiber. So now we're going to go to contraction. So for step 8, for contraction, we have to look closely at this protein here. So this protein is called actin. So this protein with 2 rows of beads is called actin. Now the thing about actin is it has little active sites on it, okay? So it has little spots on these actin molecules, okay, where these myosin heads here, these little myosin heads, these little golf club heads, they want to bind to those spots on the actin. But in the normal state of affairs, those little heads on the actin, they are covered by a protein called tropomyosin, okay? So there's this protein along here called tropomyosin. And it blocks the active sites on actin. And the reason is because we don't want the muscle to be contracting all the time. We only need it to contract when we need it to contract. So when you don't need it to be contracting, you want that tropomyosin sitting there, keeping the active sites shut down, basically. And in addition to tropomyosin being there, there's another protein involved called troponin. And troponin is like a little latch, okay? And it's this like latch on the tropomyosin that keeps the tropomyosin in place, okay? So it's not enough that we have the tropomyosin there. We have to have the troponin. So let me point to this one and put troponin. We also have the troponin that locks tropomyosin in place. We know we have the active sites on actin. If we want to contract, we need those active sites to be exposed. But in the meantime, when the muscle is relaxed, tropomyosin is covering the active sites, and troponin is in place, acting like a little latch to lock down the tropomyosin. We spent all this time earlier trying to get calcium released, right? Remember here we released calcium? So why did we release calcium from the sarcoplasmic reticulum? Well, it turns out calcium is the key that opens up that lock, that opens up that troponin. So what happens is calcium ions are going to bind to the troponin, So we're going to get calcium binding to that troponin. And when that calcium binds to that troponin, it causes tropomyosin to rotate out of the way, okay? So basically, tropomyosin undergoes a conformational change, and it rotates out of the way, exposing the active sites on actin. So all of that is step 8, okay? So step 8 is calcium. So here's the calcium, binds to troponin here, causing tropomyosin to undergo a shape change and rotate out of the way, exposing the active sites on actin. Again, these two rows of beads here, that was actin. Okay, so now we've exposed the active sites. So that's great. So now what? So now we need to set some things up for step 9 before we deal with the active sites. So let's go down to our myosin heads down here and let's put some things in place. So we need a circle here and a little circle here. We're going to need a big circle here. And we're going to need a little circle here. And then we're going to need a big circle here. And then we're going to need a big circle here. Okay, so what these things are down here, these like little golf club looking things, these are your myosin heads. So this is your thick filament, your myosin proteins. So what myosin heads have on them is the myosin head has a molecule of ADP, which is like ATP but one less phosphate, and it has an inorganic phosphate on it, okay? So ADP and PI. So what happens is here, the myosin head is cocked and ready to go. So for number 9, what's happening is the myosin head is cocked and ready to go. It has ADP, it has a molecule of ADP and a molecule of inorganic phosphate on it, and it's cocked and ready to go. So what happens, as soon as those active sites are exposed, it knows exactly what it wants to do. It goes shoop, and it locks right in. It locks right into that active site, okay? So a bunch of myosin heads are gonna lock right into those active sites on actin. And when that happens, that's called a cross bridge, okay? So the cross bridge forms between myosin and actin, okay? So as soon as the active site opens up, that myosin head is sitting there cocked and ready to go. It's got that ADP and that inorganic phosphate on it. It's ready. It's just waiting until that active site opens up. That's all it's doing all day. As soon as that active site opens up, it's like shoop, and it locks right in, okay? Now, what's going to happen in step 10? We still have the ADP here, and we have the inorganic phosphate. But if you notice, so the inorganic phosphate here, PI, it takes a hike, okay? So the inorganic phosphate leaves the head, and when that happens, the myosin head holds on tighter to that active site. So myosin head holds on tighter. Okay. So now that myosin head is still locked into the actin, but now it's holding on tighter, okay? And the reason it needs to hold on tighter is because it's getting ready for the power stroke. It's going to essentially bend, and it wants to make sure it's holding on real tight when it does that, so it doesn't let go of the actin. So in the next step, what you'll notice is over here, now the ADP leaves. So the ADP leaves, and when the ADP leaves, what you notice is the head goes from being in its original position to essentially it's called the power stroke, like flexes a little bit, and that pulls on the actin molecules and essentially shortens those sarcomeres. So you're pulling from the right and the left, and it pulls that sarcomere, and that allows the whole myofibril, which in essence is the whole muscle, is going to shorten, okay? So the ADP leaves, causing the power stroke. So that's going to be 11. ATP leaves, causing the power stroke. That is the moment of contraction. That's when your sarcomere shortens. So myosin pulls on actin. Myosin pulls on actin. Sarcomere shortens, okay? So this is the whole thing we've been leading up to, the power stroke. That's your contraction. That's your moment of contraction. And then after that contraction happens, a new molecule of ATP is going to bind to that myosin head. So that's going to be step 12. A new molecule of ATP binds, and that's what's going to allow the head to release from actin. So this entire time, ever since that active site opened up back here, right? Ever since that active site opened up, that myosin head has been locked into that active site. So through step 9, through step 10, through step 11, that myosin head was bound to that actin. But as soon as a new ATP molecule binds right here, now that myosin head can be released from actin, okay? So a new ATP binds to the myosin head to release it from actin. So new ATP binds to myosin head to release it from actin. Okay. And then immediately, just chemically, that ATP is going to split into ADP and PI. And so you're back at step 9, basically. Like now you're sitting cocked and ready with the ADP and the inorganic phosphate on the head, ready to go for the next round. Okay. So all that, 8, 9, 10, 11, and 12, those all together are going to be considered contraction. And again, that contraction part happened here at the power stroke. That's when the actual sarcomere shortens. When the sarcomere shortens, the power stroke causes it, and it causes the sarcomeres to shorten. That is your actual contraction moment, when the muscle contracts. Okay, and then finally, we get to relaxation. So the couple of things we did is we dumped all this calcium into the sarcoplasmic reticulum, so we need to do something about that. And we dumped all this acetylcholine into the synaptic cleft, so we need to do something about that, okay? So number 13 let me get a different color here. So for number 13, I'm just going to put it next to this calcium here just so we don't confuse the issue. For number 13, we need to pump this calcium back into the sarcoplasmic reticulum. So pump calcium back in to SR. So we need to pump the calcium back into the SR in order to have enough calcium there for the next contraction, because we know that calcium being dumped out of the sarcoplasmic reticulum is essential in order for the sequence of events to take place. So one part of the relaxation phase is pumping the calcium back into the SR, and the other part of the relaxation phase is there's an enzyme called acetylcholine esterase. So I draw enzymes as Pac-Man. I think I told you that before. So the acetylcholine esterase is going to come in and break down that acetylcholine and recycle it back into the neuron, okay? So this little enzyme here is called acetylcholine esterase. Like I said, it's going to come in and it's going to recycle. It's going to break down that acetylcholine so that it can be taken back up by the neuron and used again for the next muscle contraction. So that's going to be the other part of 13. The other part of relaxation involves the breakdown and reuptake of acetylcholine. So we have this reuptake of the calcium ions into the SR, and we have the breakdown and reuptake of acetylcholine into the neuron. So both parts of 13 are going to be called relaxation. So that's the story there. 14 steps broken down into 4 sections. So those 4 sections are stimulation, excitation, contraction, and relaxation. And so then in each one of those steps, you just break it down into basically its sub-steps. So we need to know a lot of those details going into lecture exam 3. Obviously, there'll be a homework on this. We'll go over it again in class when we all come back after Thanksgiving, but there is your first preview into it.